Good afternoon. I'd like to thank the organizers and particularly André Esposito for kindly inviting me for this magnificent meeting. As a professor of this institution, I'm very proud of having this meeting here, and I hope in the near future the organizers are kind enough to bring At the Limits back to Campinas once more. Well, obesity has grown very fast. Its prevalence is growing very fast over the last 40 years in the whole world. And currently, we have approximately 650 million people with obesity in the world. Projections say that within 6 or 7 years, we'll probably reach the number of 1.2 billion people with obesity. Well, you all know that obesity is the main risk factor for all the diseases we are talking about in this meeting. So it's very important that we are capable in the near future to achieve pharmacological advance that we can use to prevent and to treat obesity properly. Obesity is the simple result of overeating and physical inactivity. It's quite intuitive to think that if we are capable of developing strategies to reduce food intake and increase physical activity, patients should lose weight. And that's true. If we do that, and if we achieve these goals, patients do lose weight. However, most clinical studies using only behavioral approach to treat patients with obesity have shown that, in general, we achieve only 5% body mass loss, and the worst is that patients are not capable of maintaining the body mass that they have lost. So obesity is a very recurrent disease and very difficult to treat because of this recurrence. Well, we have two distinct neuronal systems that control food intake, and controlling food intake is very important in order to achieve the goal of leading to body mass reduction. So we have a homeostatic system, which is in my right-hand side of the slide, right here. The homeostatic system responds to hormones and to nutrients that reach the brain to inform the amount of energy that is available in our body. And the neurons respond to these signals by controlling food intake and energy expenditure. On the other side, we have the hedonic system that responds mostly to visual, olfactory, and gustatory stimuli, and it's very much regulated by social situations, such as, for example, going out to eat with your friends or partying, etc. Under physiological conditions, the homeostatic system is capable of maintaining the hedonic system under control in such a way that when we overeat, for example, after a period of overeating, we reduce a little bit the consumption of foods and we return to body mass stability. A very nice example of this in real life is this paper published in the New England some few years ago. They have evaluated body mass change over time, over a 1-year period, in 3 countries, in Japan, the United States, and in Germany. As you can see, body mass increases very much during Christmas time in all countries. And then you have some particularities about body mass increase. For example, in the United States, you have Thanksgiving, United States in the yellow color. In Germany, you have Easter, which is very important for the German culture. And in Japan, there is a golden week. They eat quite a lot in this time. But as you can see, in all times when body mass increases after a holiday like this, it goes down. That's for the majority of people. So this is an example of overeating because of overactivity of the hedonic system under social condition. And then the homeostatic system comes afterwards and reduces body mass to stability. So this is what happens under physiological conditions. However, under non-physiological conditions, and there are a number of examples of what we can say what is a non-physiological conditions regarding food intake, but I give you one example: overeating of highly energetic and energy-dense foods, which is consumed by a number of people in the world nowadays. So under these non-physiological conditions, I'll show you that the neurons of the homeostatic system becomes damaged, and they can no longer control the hedonic system, which overimposes and leads to body mass gain continuously. So the damage of the neurons of the homeostatic system is a very important component of the obesity and one of the main reasons why obesity is so recurrent. Well, the homeostatic system is mostly based in neurons in the hypothalamus. And the identification of leptin in 1994 was one of the most important advances in this field that allowed the identification of distinct neuronal subpopulations that respond both to hormones and to nutrients. I'll go through some of this current knowledge. So if we take a close look in the hypothalamus, so what you see here is the most ventral part of the hypothalamus. This is the third ventricle, and this is the medium eminence. The medium eminence is out of the blood-brain barrier, whereas the arcuate nucleus, which is the most ventral part of the hypothalamus, is within the blood-brain barrier. However, the blood-brain barrier in this region is quite leaky in such a way that nutrients and some hormones can cross the barrier and signalize to these neurons what is going on in the body. So we have a number of hormones that can cross this barrier and activate or inactivate distinct subpopulations of neurons in the hypothalamus, leading to a control of food intake and energy expenditure. One important neuron in this context is this POMC-CART neuron. It is active when there is an energy surplus in our body. And leptin is the main controller of this neuron, as well as of this neuron, which is active when we are fasting for a long time, for example, or when energy stores in the body are low. So the correct balance between these two subpopulations of neurons is very important to maintain body mass stability, and that's exactly what is lost in most obese situations. Well, when leptin was identified, many researchers thought that it could be a good target for treating obesity, and certainly many of you remember there were some clinical trials using recombinant leptin to treat obese patients. However, this was a big failure because, in fact, most patients with obesity, they have very high levels of leptin in their blood, and they do not respond to additional increase in leptin levels. This led to the definition of a resistance to leptin in the hypothalamus. So we currently work with the concept that obese patients and also obese models of obesity, which I'll talk a little bit later in the presentation, they do have a hypothalamic resistance to leptin in such a way that even under very high levels of leptin, they cannot control properly food intake, so they have increased food intake, and they cannot control appropriately energy expenditure, so they have a reduced energy expenditure. And that's an important reason why they become obese, and they are always recurring in obesity when they are treated by different types of methods. Well, some years ago, more than 15 years ago, we started to ask in our lab, what is the basis, what is the anatomical and molecular basis for the dysfunction of the hypothalamus in obesity? And for doing so, we used animal models, and I think it's quite reasonable for you to understand, to study hypothalamus, we need animal models because we will never get a piece of human hypothalamus, for example, to study receptor activation, cellular abnormalities, and so on. So we have to rely on animal models. And in this study, we evaluated changes in gene expression in the hypothalamus of an obese model of diet-induced obesity. So the rodents were submitted to a fat-rich diet. They became obese. We extracted the hypothalamus, and we evaluated using a gene array a number, thousands of different genes that were expressed in the hypothalamus. And what we found is that obesity was accompanied by a 15% change in the expression of several genes in the hypothalamus. When we analyzed these genes by functional cluster analysis, we could see that most of the genes undergoing modulation, they were related to inflammatory functions. For example, the cytokines I show you here. What happens is that when the hypothalamus is chased with the fat present in the diet, it starts producing the cytokines, and the cytokines will act upon the neurons, the same neurons that I showed you a few slides before, that are responsive to insulin and leptin. So it explains why the neurons becomes resistant to insulin and leptin. Well, we then asked which components and which cells of the hypothalamus, which components of the diet and which cells of the hypothalamus were involved in this inflammatory response. And we could see that the long-chain saturated fatty acids in the diet were capable of activating TLR4 receptors in the microglia, and the microglia are the macrophages of the central nervous system. So the microglia responds to the increased presence of saturated fatty acids in the diet, produces the cytokines, which will act upon the neurons to produce insulin and leptin resistance, and therefore obesity. A number of other labs working in the same field were capable of identifying distinct components of this inflammatory signaling in the hypothalamus in such a way that we can say nowadays that in experimental models of obesity, there is an inflammatory activity which is localized in the hypothalamus, leading to a malfunction of the neurons that control food intake and energy expenditure. One important aspect of the lesions that occur in the neurons of the hypothalamus is that it is very similar to the lesions we see in neurons in conditions associated with aging. So, for example, researchers have seen mitochondrial dysfunction in hypothalamic neurons, defects in autophagy, defects in the function of the endoplasmic reticulum, defects in protein processing, and RNA stress granules. They are all common to diet-induced obesity and aging, and they lead to a deactivation of an inflammatory cascade, finally leading to neuronal dysfunction in the hypothalamus. Well, all these studies were performed in animals, as I told you, and of course we want to see which kind of abnormalities we can see in the human hypothalamus, but that's very much difficult because, as I told you, we cannot get a piece of the human hypothalamus, at least not a living human hypothalamus. So we started some collaborations with neuroscientists to use functional magnetic resonance to study the hypothalamus of humans, obese humans, and this was the first study performed in our group. And here, what we evaluated were a group of 13 patients submitted to bariatric surgery. They were submitted to a functional magnetic resonance before bariatric surgery and after bariatric surgery, approximately 8 months after bariatric surgery. They had lost approximately 40 to 50% of their overweight. And the question we went to ask with the functional magnetic resonance is if there is any change in neuronal activity on a given region of the brain. In that case, we wanted to see the hypothalamus. So the main finding of this study is summarized here. So we had a group of lean subjects, our controls, the obese subjects before surgery, obese subjects after surgery. When they enter the machine, the magnetic resonance machine, they are all fasting, and we start making acquisitions. And as you can see, all three groups have similar neuronal activity as measured by the functional magnetic resonance method. Then they drink a liquid with 75 grams of glucose, and the exam continues. And as you can see, all groups have a rapid and sharp increase in neuronal activity. The lean subjects can keep this activity for a long time. This is approximately 20 minutes exam. And the obese patients, before surgery, they have a very rapid drop in their signal. And after surgery, there is something in between. So how do we interpret these signals? We interpret that as a signal of satiety. The patients were fasting in the beginning of the exam, then they have 75 grams of glucose, which rapidly reaches the hypothalamus. The neurons detect the presence of glucose, which is nutrient, and start firing for presence of nutrients. So what we can see here is the big difference that exists between lean subjects and obese subjects. It seems to be some recovery after some time in the obese patients that were submitted to bariatric surgery. Well, other groups have been doing fantastic studies in the same field using distinct methods of magnetic resonance. This is a very good example, a group from Seattle in the USA. And they use another method of magnetic resonance to evaluate the degree of gliosis in distinct areas of the brain. And what they could see is that there is a correlation between body mass index and a signal of gliosis in the hypothalamus. And they have several controls for other brain regions where they cannot find this kind of abnormality. So we have some good work done in this field, and we can say that there are signs both of abnormal function in the hypothalamus of obese humans and also structural abnormalities. So we have started a collaboration with the Seattle group to study changes in the hypothalamus of children. As you know, yesterday we had a magnificent talk about about cardiovascular problems in the young population. And of course, you know, the prevalence of obesity in children is increasing rapidly, and that will certainly be a problem for us in the future. So we designed a study together with the Seattle group to evaluate the hypothalamus of 12 obese children in comparison with 11 lean subjects. And we used both the fMRI, the method that we used here, and the structural method. I'll show you the structural because of time. So this is the result of the structural evaluation. As you can see, the obese children have increased signal of gliosis in the medium basal hypothalamus, but they have not in other control regions of the brain. So children with 12, 13, 14 years of age, which are obese, have already signals of abnormalities in their hypothalamus. And this is correlated with body mass index as it is in adults, as I showed you. Okay, how do we overcome hypothalamic abnormalities in obesity? Well, differently of the incredible pharmacological progress that we've seen during these 2 days in cardiovascular disease, in hypertension, and in diabetes, in obesity, we are just in the beginning of understanding the pathophysiological mechanisms leading to the recurrence of obesity, so we are still looking for good targets. And I'll show you the story of one good target. That's certainly the beginning of our increase in pharmacological advance in obesity. So GLP-1, as you know very well, the GLP-1 receptor was a good target used initially to treat diabetes, but both in preclinical and clinical studies, body mass reduction was always observed. So researchers start thinking, well, it probably acts in another way other than insulin production. So they started looking for potential targets for GLP-1 in the central nervous system. Here is an example of what you can get of body mass reduction with a GLP-1 agonist. So this was a very interesting paper that was published in 2014, and the great advance in this study is that the researchers could label the liraglutide molecule, the GLP-1 agonist molecule, with fluorescence and treat experimental animals and see exactly in what parts of the brain liraglutide was binding. And to do that, they used a method, a chemical method, to make the mouse brain transparent. So what you see here is a mouse brain. This is the cerebellum, this is the cortex, and it is transparent. This mouse was treated with fluorescent liraglutide, and as you can see, most liraglutide binds to the hypothalamus. And we have a close look on that. You see that most of it binds exactly in the arcuate nucleus, the most ventral part of the hypothalamus, exactly where the neurons that control food intake and energy expenditure are located. Here, they used a recombinant mouse that expresses POMC, one of the neurotransmitters involved in food intake and energy expenditure. It expresses POMC in green, so all neurons expressing POMC are green. And then they labeled liraglutide with a red fluorescence. And as you can see, liraglutide binds to POMC neurons. It does not bind to the NPY neuron. I'm not showing here, but it binds to POMC neurons. And they have proposed a mechanism of action for GLP-1 agonists in the central nervous system. So it binds to POMC neurons. It binds to another neuron that is not completely identified. It's a GABAergic neuron. And this neuron provides a negative feedback to control NPY. So we have a positive stimulus to POMC, which reduces food intake, increases energy expenditure. And we have a negative stimulus to NPY, which does the opposite. So this is very nice because we begin a new era of obesity treatment with a drug that we know very well how it works at the cellular level. Well, during the process of mapping the actions of GLP-1 agonists in the brain, a number of interesting papers were published, and this one is very nice because they used both a histological analysis of the stimulus provided by GLP-1 agonist and also gene expression response. And then they could map the primary sites of action of a GLP-1 agonist and the neurons that are in second order, so the ones that respond to the primary sites of action. As you can see, they confirm here that most of the primary binding sites for liraglutide is exactly the hypothalamus, and they can activate a number of regions of the hypothalamus. But then, when they looked for the secondary, the second order neurons, the ones responding to the primary order neurons, they could see that there was a stimulus to neurons located in the limbic system, exactly the region where you have hedonic control of food intake. So to finish my presentation, I'll show you one study that explores exactly the question of whether we can have changes in hedonic feeding when we treat patients with GLP-1 agonists. To understand this experiment, it's very important that you take a look in here. So the patients go to a functional magnetic resonance scanning with a glass that projects images. So the researcher can choose which kind of image is going to be projected, and I'll show you some examples of that. So you have, for example, high-calorie food pictures, neutral pictures, and so on. During the whole process, the researcher asks to the patient what is the feeling of that, what that image causes as a feeling, and the patient responds to that question, and there is a score of emotional eating, which means that you see the food and how much you get excited about that food. So let's see what we can get out. So the patient goes inside the machine with the glass, projecting images like this is a neutral picture, then an interval, then a picture like this, and the patient goes on answering to the questions, and then you have first question, is there any brain signal related to emotional eating? Yes, there is. in the amygdala, one of the components of the limbic system, and one of the regions where we have neurons responding to GLP-1 agonism. Well, very good. So the second question, does GLP-1 agonism modify neural responses to food? And then the experiment is changed a little bit. So there is two parts in this experiment. First part, the patients are fasting. They go to the machine, they are exposed to the images, and the researcher calculates the emotional eating. Then the patient is given the food, the nice food that was projected. He eats that, and he goes back to the machine, and then we can see the signal again. So what we have here is that before food intake, there is a very high anticipatory food reward signal, which is in the amygdala again. And here the patients are under placebo. Afterwards, when the patients have consumed food and they go back to the machine, then there is a negative signal. So it means that the patients are very much more excited about seeing food than eating food. And that's very common in obese patients, because they need to eat more in order to get the same kind of pleasure that they think they will have when they see food. However, when they are treated with a GLP-1 agonist, the situation is completely reversed. So before food intake, there is no big signal. However, after consumption of food, the signal is good. It means that eating the food gave you the pleasure, and you can see that in changes in neuronal activity in regions of the brain which are related to hedonic feeding. So I want to conclude saying that we are starting to understand how food intake is controlled. There is a long way to go. We are starting to find good targets to treat obesity. We still do not have this tremendous pharmacological advance we have in other related fields. But of course, if in the future we have drugs that can properly treat obesity, we will have a tremendous impact in cardiovascular disease, in diabetes, in hypertension, and so on. So I want to thank you very much for the invitation and for the opportunity, and to come here to our town and to our university. Thank you very much.